Hello, everyone. In this video, I'm going to show you some of the PDB web pages and the kinds of annotations that are available on them, and PyMol, how to use PyMol, and then a few web pages where we can take a sequence and investigate some of the properties of it, as well as the Unipro pages. Occasionally, I will say something like, you could pause here. If I've shown you a bunch of things in PyMol, then I may say pause. You can hit the pause button, and then you can do those things in PyMol on your computer. And then when you're ready, come back to the video and start it up again. Because normally when I do this in person, I can do stuff on the screen, and then a couple of my students usually come with me and walk around the room and see if see that everybody's managed to figure out how to do a particular step. And we sort of do it, back and forth like that. But in these circumstances, we don't have that option. So let's see how this works. I'm going to hit the share screen button this time. And then I'm way up here. Make that a bit bigger. Okay. The sort of outline that I'm going to follow is, if you go to dunbrack.fccc.edu/T temple, the bunch of files in here, including the talk from Tuesday, which is this BMSC8203 dunbrack1.mp4. The HTML page will stream it so that you don't have to download the whole thing, but you can also, if you click that, you would be able to download it, as well as the slides. Down here is temple_march2020.html. And I'll click that now. That is basically the outline of what we're going to go through. It has lists some of the commands and things that I'm going to show you. I have it open on another screen here, so I'm just going to follow it on my old laptop. And show you things on the screen. But you could use this page to kind of go through things again after the video or even during the video. So let's get started. We're going to go to the Protein Databank. And the Protein Databank is www.rcsb.org. There are actually 3 sites for the Protein Databank. This is the site in the United States, up at Rutgers University. And the other sites are PDBE in Europe and PDBJ in Japan. I think pretty soon there's going to be one in China as well. Let me make this a little bit smaller. Put it like right there. So we're just going to look through, for a couple of PDB entries, some of the information that is in a PDB page. So up here, if you know the PDB ID, let's say you read a paper and it mentions this has been deposited in the PDB under this code, then you can just type that in here. You can also search by protein name, by organism. There's an advanced search. Actually, if you go to beta.rcsb.org, beta.rcsb.org, and then go to search, they've revamped the search function, and somewhat soon I think that's going to become the regular search pages. So you might take a look at that. I'm going to put in 4ZY4, and remember that a PDB code begins with a number and then is followed by 3 letters or numbers or combinations of. The PDB will suggest a hit. You can just click on that or hit return there. And that will go to PDB ZY4. I'm going to make it a bit bigger. If I make it really big, it'll actually switch formats to something like that. You can't see the whole page that way. So let's just kind of go through what's on this page. You get things like the title, some classification for it, the organism that it's from. This is the PAC1 kinase domain of the human PAC1 gene. And if we scroll down, we get the literature, and you can click on the PubMed ID You can actually download the citation if you want to put it in a paper, download EndNote or Mendeley. It's very important actually to cite crystal structures, not write or experimental structures. You don't want to just put the Pdb code in a paper. You actually want to cite the authors. They did a lot of work. They deserve a citation when you show a picture of their structure or discuss it. If we go down a bit further, we get to what's the contents of this PDB entry. And so the first section is the macromolecules. This will be the proteins or nucleic acids, what the protein is, the species. You'll get this kind of domain, this kind of view of the whole sequence. So where it says Q13153 here, this is the entire PAC1 sequence, this sort of gray bar. What you see down at the bottom, these blue bars, this is the part of the structure that was expressed by the authors. They made in vitro and purified and made into a crystal, in this case, a crystal structure. Because they didn't express the entire protein. That's very common that people express certain domains and not others when they're doing a whole structure. You can also see there's a little break right here, sort of a little section in gray, right, in that part of the sequence. What that indicates is that they were not able to model the coordinates of that section. What happens in crystal structures quite frequently is some loop will be moving too much, and you won't see any electron density when you get to modeling, calculating the electron density. And so crystallographers will just leave out the coordinates for those residues. And that's very important. You want to, it may be that the structure of that loop or that region is very important to you. And so you need to go find a structure that has that piece. And in fact, we can do that if we see where it says full protein feature view for Q13153, that's PAC1. If I click that, I get a page on Q131 on PAC1. And down here, you see it's basically the same kind of layout, except it has all of the PDB structures of this particular protein. I have to hit the plus sign to get the full list. And so these are all that same kind of bar chart. So let's say I was interested in, here's 4ZY4, here's that missing piece. If I scroll down, if I look down a little bit, I can see in 4ZY6, that piece is present. The tricky thing is that sometimes we look at 4ZY4, where my cursor is, the rest of the structure is there, but if I look at 4ZY6, chain B, where'd it go? Right here, there's another piece, this piece of the activation loop that's missing. So you can try to look around for a complete structure. Here's one that appears to be complete. These are these little lollipop things are usually mutations or covalent ligands or other kinds of variations in the structure. And again, you get different kinds of annotations. You get the PFAMs. PFAMs are protein families. They're, this region is the protein kinase domain. This is the binding domain, PAC1. You can even get information on exons and the gene, things like that. So I'm going to close that up again, go back to the other page. If we go down a little bit further, we get to the small molecules, any kind of ligands that are in the structure. And very frequently we crystallize proteins, we get ligands from the buffer that's used to crystallize the structure. And so here we would have sulfate ion, dimethyl sulfoxide, a picture of it. If I click one of these links that says ligand interaction, it will actually show the ligand in the structure. I guess I can do that really quickly. The ligand. And in a viewer, you can actually see, I think I'm using 2 fingers to scroll inwards here. You can zoom in and see this is the drug that is bound to the kinase. These are some of the side chains of residues that are in contact with it. That's the full list. If you click on one of these ligand identifiers, so amino acids are given in a 3-letter code, the standard 3-letter code. Ligands are also given a 3-letter code. So every unique ligand in the PDB has its own 3-letter code. So I could click on 4T3 over here, and that would open up a page that has more information about that ligand. All sorts of chemical properties and the hash strings associated with those. There's some experimental data. One of the important things on the web page, I guess it's here at the top, and you can also see it down here at the bottom. Let's go to the one at the top. Is this what says WWPDB validation? WWPDB means the worldwide PDB. It means all 3 organizations together. And we click that. That actually downloads a PDF that has a lot of information in it if you want to get more details. But let's just take a look at this, this little image right here. What the PDB does is it tries to evaluate the structure in a number of different ways so that you can tell whether this is a good structure. And if you're comparing 2 structures, you might want to compare this little bar chart for one PDB entry and another PDB entry to see which one is the better structure. A couple of those features, the R-free and the RSRZ outliers, have to do with the electron density and how well the coordinates fit into the electron density and how well those coordinates predict the experimental data, the structure factors, the intensities of the spots in the diffraction pattern. So there's a value called R-free, which means you hold 10% of the spots to the side and then basically determine the structure with the other 90%, and then calculate, well, what's the value of those remaining 10%? And then calculate them from the coordinates and then compare them to the experimental values for that 10%. And you can do that over and over again, different groups of 10%. The authors, so if we look a little bit to the left of the validation, the authors have said what the resolution is. They have said what the R value, the free R value is, and what the work R value is. So the free R value is taking 10%, putting them aside, and what's the value on that 10%. The work is on all of them. The R free is the one that is the relevant one here. And it basically says there's a 25% disagreement between the predicted structure factor values and the experimental ones. That's actually not bad. That's a sort of fairly normal value. And so what you see in this, for all of these things, is this sort of bar, this little bar chart. And the ones to look at actually are not the ones that are connected by lines and the filled ones, but these empty boxes here. And it says in very small type here, it's bigger. Percentile relative to X-ray structures of similar resolution. So the resolution of this structure is 2.6 angstroms. Just to give you a feeling for resolution, above 3 angstroms, a number larger than 3 angstroms is a pretty low resolution structure. Can't really get all of the side chains correctly oriented. Ligands might be not very well modeled. Between 2 and 3 is a sort of decent resolution. You can see most details. 1 angstrom to 2 angstrom are fairly high resolution structures, especially at the 1 angstrom end of it. But there aren't many structures of that high resolution. So typically, I'm happy if I can get a 1.8, 2.0 resolution crystal structure. But sometimes you don't have any choice. The only structure available is 2.8 angstroms, and it's not a very good structure. What the PDB does is it calculates R-free on their own. They calculate R-free. And so that's the value that is here, 0.248. And that's exactly what the authors said it was. I have seen cases where the authors say it's 0.25, but the PDB calculates 0.4. And a 0.4 R-free is that's a terrible structure. There's something grossly wrong with it. The one example I can think of they actually modeled the wrong protein sequence into the structure. It's kind of a long story. If you want to look at that page, it's 1OCW, the letter O, 1OCW, and you can see what I'm talking about. So this one has 0.248, and the blue end means good and the red end means bad. So the fact that this is sort of towards the blue end means for this resolution, this is not a bad value. The RSRZ outliers is how well the atom fits the local electron density, and how many atoms are maybe misplaced. They're either in low electron density, or where there is electron density, they didn't put an atom, things like that. And this structure actually is not so good with that. It's on the low end, both for its resolution and all X-ray structures, which are the filled bars. The other three criteria are structural, geometrical. They are, remember mole probity that we talked about in the Tuesday lecture. If you go to that website, you get more detailed information on these values. The clash score says, are there a lot of atoms that are just too close together? Non-bonded atoms, aren't covalently bonded to each other, at least directly, that are overlapping a bit. And if the structure is well modeled, you shouldn't have any of those. And so this structure does okay with that. Ramachandran outliers, as the name implies, are how many of the residues are outside those contours in the plots that we looked at yesterday. And the same thing for the sidechain outliers. How many rotamers are either very rare rotamers altogether, or the dihedral angles are not even rotameric when they should be. So a structure that had a 120-degree dihedral angle for chi1 instead of 60 or 180 would be considered a sidechain outlier. And it's just done as a percentage of all of the sidechains, 3.3%, which is not too bad. If we look on the left-hand side over here, the PDB webpage always opens up on the biological assembly. We talked about those, meaning what's in the crystal that the authors think is the most biologically relevant structure complex that's inside that crystal. In this case, in 4ZY4, the authors have said the biological assembly is this monomer. They're actually wrong about that, but that's another story. And so they say the global symmetry is asymmetric. It's C1 because it's just a monomer. The global stoichiometry is monomer, or A, in that format, that A2B2 type format. And if we click this forward button, we can see that there's a second biological assembly, which is also a monomer. And if we click it one more time, we will get to the asymmetric unit. So the asymmetric unit is actually a homodimer. And it says Homo tumor, or A2, and it's asymmetric. This is actually one of the examples of an asymmetric homodimer. In fact, this is actually the biological assembly. This would, if they had noticed it. This is a kinase where the activation loop of one of these monomers is sitting in the active site of the other one, as if it's about to get phosphorylated. And it appears to be, the mechanism for phosphorylating, for one kinase phosphorylating the structure of another copy of the same kinase. And that's an asymmetric situation because one is acting as an enzyme and the other one is acting as a substrate. But we can hit the button again and we get back to biological assembly 1. Let's see, anything else on these pages? There are a bunch of annotation tabs up here, and you can click through those just to see what's on them. I'm particularly going to look at the one that says sequence. And on this page, we get this kind of logo of the sequence, where the amino acid sequence is listed here. If you mouse over it, it will tell you the residue number. You can see the residue number kind of pop up. And the view that you get, what you get is this line that says DSSP. And DSSP is the secondary structure. DSSP is a program that you apply to a set of coordinates, and it tells you there's a helix from this residue to this residue, there's a beta sheet from this residue to this residue, there's a beta turn that we looked at in certain positions. And so the wavy lines are the helix, the arrows are beta sheets, They're not oriented parallel, anti-parallel, all of that. They're just from the end terminus of the sheet is the blunt end, and the C terminus of the strand, beta strand, is the arrowhead. These little green kind of curves are where beta terminals are. And there are other kinds of annotations. These little yellow circles are site. It says site record. That means that it's a residue that maybe is binding to a ligand, something like that. One important thing to notice is where there's a break in the DSSP line. So there's no DSSP line up here. There's also no residue numbering down here. That means these are residues where there are no coordinates. And in fact, if I leave my mouse on it, it says no identifier from atom record. In other words, there's nothing in the atom records, the coordinates, for this N, this asparagine residue. And so this is another way to get a quick look at the sequence, some features about it, and where the disorder is, and exactly which residues are missing. You can also add annotations. For instance, you can, PFAM is this set of different protein families, a kind of a nomenclature for different protein families, and I've just added that, and it says PFAM, and it says protein kinase domain, which covers most of this, the sequence that's in this entry. Okay. There is a 3D view page. So the PDB has its own software for viewing structures, and you can play with these menus. I think PyMol is easier to use, and that's what we're going to show. I'm going to show you today. Their newer one is called Mol Star, and I think this will probably become the default. The nice thing about it is actually it has the sequence up here, and so you can click and select residues, which you ought to be able to see on the structure. Yeah, there they are, sort of highlighted. And that's nice, because otherwise it's sort of hard to navigate between the sequence and the structure. And in the older viewer NGL, you don't have that option. Okay, I think we went through everything on my list. This is the beginning of that HTML page I showed you. So now we're gonna look at a couple of other examples of how the biological assemblies are annotated. And so we're gonna go to PDB entry 1M54. This is an enzyme called cystathionine beta synthase, CBS. One of my colleagues, Warren Kruger, works on it. And it says the biological assembly is a homotumor. It's cyclic. Remember, so this is a cyclic homodimer, which is an isologous interface. And there's a picture of it. And if I hit the arrow key, there's another one that's also a homodimer. If you looked at these together, you would see that they're basically the same homodimer. I'll explain why in a second. I think there's a third one. And then finally, we get to the asymmetric unit. And the asymmetric unit has 6 chains in it. And so what the authors have done here is they've said the biological assemblies are only homodimers. And it turns out in the asymmetric unit, there are 3 copies of that homodimer, each with slightly different coordinates, because they've modeled it as a homodimer, and they sort of had to in order to get a good model of the crystal. And so what the authors do is they take the coordinates of basically of the asymmetric unit and break it into 3 separate biological assemblies that basically look the same. You might want to look at them. Maybe there's a loop that's present in one that has coordinates in one of them that is missing in the coordinates of the other. If you go to the sequence tab, I think you have to hit a button that says show all chains, and you can look at all of the chains and see, are there coordinates for the amino acids that I'm interested in, and which chains are those present in. Right. If we open up a different PDB entry, I'm going to go to www.rcsb.org. And I'm going to put in 2Q3B. This is also This is a protein that's related to CBS, actually homologous protein to CBS, and it has the same homodimer. And there's the biological assembly, and there's the asymmetric unit. The asymmetric unit is only a monomer. The stoichiometry is just A. And so what the authors have done is they have used the symmetry operators to build a second copy of the protein, and that's what's in the crystal. If you look at the crystal as a whole, you could see this dimer makes up the crystal. And then finally, one more example, 1LCY. And this one says that this is a monomer, the biological assembly. If I hit the button, I should see biological assembly 2, which is a trimer. And at the bottom here, it says biological assembly 2 generated by PISA and PQS. Remember, PISA is a software that can look at a crystal and kind of hypothesize or sort of predict what is the correct assembly inside that crystal. And the PDB adds these annotations because they are different than what the author put a monomer, but PISA's relatively confident that it's a trimer. And so the PDB provides you with the coordinates of the trimer. What's sort of interesting, I think, if I remember right, if we look at the abstract here, the abstract says this is a pyramid-shaped Homo trimer. So what happened was the authors know that it's a trimer. They showed a picture of the trimer in their paper, but when they deposited the biological assembly, they only put the monomer. They just copied the asymmetric unit. And this happens, as I said, about 10 to 20% of the time, where there's either a miscommunication, the authors didn't know what they were being asked for a biological assembly. I think that's better now. I'm on the PDB's advisory board, so I get to request that they change things. And so now they show authors the different PISA biological assemblies and say to them, pick one of these, unless you think it's something else, and then give us the coordinates for that. But otherwise, here's a picture of it. Oh, and most authors will say, oh, there's the trimer. Yeah, I'll pick that one. So this is a fairly old structure from almost 20 years ago. You can also see that it has poor class scores and poor Ramachandran outliers. Some older structures don't have the experimental data deposited, so they don't have that R-free slider here, or the RSRZ slider here, because you can't calculate those if you don't have the experimental data, the structure factors. Okay. So now we're going to go to PyMol, and I guess I'll close this. I have PyMol sitting in my little menu bar down here, but what you're going to do is double click on it. Or in a Mac, you can hit this launch pad and then just type in PyMol and then click on it. In Windows, there's something similar to that. But basically navigate to it and then open up a window. And so I'm going to pause. You can pause the video here until you have it open. Okay, now I'm going to go through some of the commands, and these are listed in that HTML page as well, some of them. We're going to fetch for ZY4. So fetch is going to go to the PDB and get the coordinates, get the PDB file for the entry for ZY4. If you just type fetch for ZY4, it's going to get the asymmetric unit of for ZY4. It is not going to get the biological assembly. If you want to get the biological assembly, you have to type fetch for ZY4, comma, type equals PDB1. That seems weird, but the files, if you just ask for ZY4, the file name will be either 4zy4.pdb or 4zy4.cif, CIF. CIF is the more advanced, newer format. Newer, it's still 20 years old, actually older than that. But it's kind of the official format of PDB files. The old format was developed in the 70s, and all of the lines are 80 characters wide because in the 70s you had to program a computer and store data, you had to have little punch cards that had basically 80 character positions, and then you would put holes for the different letters or spaces or numbers, and each line was a separate card. And so they were restricted to 80 character lines, which for PDB atom, with atom, residue number, atom number, residue type, atom name, X, Y, and Z, some other information about the atom position, it's tough to fit that into 80 characters. So stuff kind of bumps up against each other. So where you don't have spaces between some of those elements, you just have to measure where they are in the column numbers. The MMSIF format is much better and is easier to work with on a computer by far. I'm going to fetch actually the asymmetric unit. And remember, this asymmetric unit for this one is a asymmetric homodimer. It is actually the correct biological one. The authors didn't notice it. I just happen to know that because we worked on this protein. But I'm going to type fetch 4zy4 and it will open. In this version of PyMol, it will actually open up in this cartoon format, and which is nicer to look like. In the old one, it would just open up as sticks and it would be too crowded. If you open up a PDB file either by fetch, or you can actually from the file menu, you can of course open up, there's actually a get PDB menu, or you can open if you have the file like sitting on your computer, for instance. You will see when you open up a new file, you will see this is called the object menu over here. And so it lists 4zy4. I can click that object menu on and off. So let's say I have, I'm going to fetch 2q0n. And now I have two objects. They're in different places, in different, if I sort of move this around, I can see where 4zy4 is. But that's, if you've got a bunch of objects open, you may want to click them on and off just so you can see only one of them at a time. I'm going to go back to 2q0, q0n in a little bit. You see that now next to 4zy4, there are 5 boxes. A is for action. It takes different, we'll do kind of different processes on the structure. S is for show. H is for hide. L is for label. And C in this little rainbow is for color. So I'm going to go to A, and I'm just going to go to orient. And it will bring 4zy4 back to the middle of the screen. And so these are relatively self-explanatory. So if I go to show and I click on spheres, now I get a sphere for every atom in the structure. And the opposite of that is I can go to hide and I can say hide the spheres. If I hide, I can hide everything. And then I can say show the cartoon. So the cartoon is this kind of representation. Again, hide everything. And I can show what's called the ribbon. It's a little bit counterintuitive because we often call that other representation ribbon. But ribbon in PyMol means this. It's the position of every C alpha in the structure. So it just follows the backbone. And every point is exactly where the atom is. Whereas the cartoon is actually kind of a bit of a schematic representation. In other words, it's kind of smoothed out. You can see, the helices are very nice and regular, but real helices aren't quite that nice and regular. The loops are kind of smooth, and real loops aren't that smooth. I will just point out, remember yesterday I said loops kind of look like this. They don't look like that. And you can see that here. So this right here would be a little beta term. we can color this in different ways. So if I go to the C menu, I can say color by chain. This is a homodimer, so each chain, one of these is chain A, and the other one is chain B. And so I just colored by chain, and that's very helpful when you're looking at a multi-chain assembly. I can say color by chain, and then I can go down to here where it says chain bows, and it will color both chains from blue to red, from the N terminus to the C terminus, from dark blue to dark red. I can pick a color. I could say I want the whole thing to be magenta. I can color by secondary structure with helices in red and sheets in yellow and loops in green. If I hit spectrum, I can color what are called the B factors. And B factors are attached to every atom in the coordinates is a number called a B factor, also called the temperature factor. And it expresses how much electron density is where that atom has been placed. If the atom is kind of moving around and kind of smeared, then it will have a fairly high value, sort of 40, 50, 60, 70, 80, kind of that sort of number, up to maybe about 100. If it's really well defined and the electron density is high as it should be for an atom with, let's say, 8 electrons, it'll be something like 10 or 20. And so you can color the structure. And what you can see is that there are some pieces of structure, like this first helix in this chain, that have high B factors, meaning that it's moving around a little bit in the crystal. And this is the activation loop of the kinase right here, and it's a little bit red. So I'm going to go back to coloring by chain. And I'm going to pause for a moment, and you should play with the S, the H, and the C menus for a little bit to get used to using those controls. The other thing to get used to is, actually before you do that, there are different ways to control the mouse or your keypad, trackpad in PyMol. You can select those up here where it says mouse, and it says 3-button motions, 3-button editing, 3-button viewing. You can read all of these. The viewing ones are the one, if you just want to look at the structure, you want to pick one of those. Editing means you might move atoms around, and most of the time you won't be interested in doing that. On my Mac laptop, I actually prefer the 1-button viewing mode. And you can see when I change that, if you look in the lower right of the PyMol window, you see the mouse, it says mouse mode, and it says 1-button viewing. And for left, middle, and right clicks, L, M, and R, it tells you what PyMol will do. And if you have a wheeled mouse, you can use the wheel. That changes if I pick a different option. I can also click right on it, and it will toggle between a couple of different possibilities. But I've gotten very used to using the 1-button viewing mode. Actually 2-button viewing. Yeah, 1-button. Either 2-button viewing or 1-button viewing. 2 is probably easier, actually, if I think about it. What it says down here is basically what's going to happen when I click, either just click or click and hold on the window. So the left says rotate, and that just means if I click and hold down the cursor, and then on this trackpad, I'm moving a second finger. So I'm pushing down, holding it, and then with this finger swiping back and forth on the screen so that I can rotate the structure around. If I right-click on a Windows laptop, typically you will have a right-click button. It says move Z, and Z is the axis, is the direction into the board. On a Mac laptop, you actually have to set what the right click is. It's in one of the settings menus. Usually it can be set to clicking in the right corner towards you on the trackpad. And so if I hold that down and then swipe with the second finger, the structure then moves in and out. And so if you want to zoom in on something, that's the way to zoom in. Right down here where it says control means hit the control key, and it says you can move the structure under the L button on a left click, regular click. And that means in the XY plane, so that means this way. And so if I hold control down with my left hand, left finger, I can, actually not working. On a Mac, actually, it's the command key, not the control key. But on a Windows machine, it should be the control key. Those are the main ones that you really need to know about. So now I am going to pause. And so what I want you to do is to rotate the structure around, move it in and out by zoom in and zoom out, rotate it around, play with the show and the hide menus. You can show surface as an interesting way to look at it. Hide those things, and the color menu. And so I'll wait for a few seconds. You can pause it. I will wait for a few seconds so that when you click start again, I'm not already talking. So go ahead. Okay, so now you'll come back to the video, and let's look a little bit further at what PyMol is showing us. One important thing that this version of PyMol and the ones previous to 2.0 didn't have is that it shows you where there are missing coordinates. So you can see that occasionally the ribbon is a dashed line. And what that means is there are residues that were not modeled by the authors. There's no coordinates for them. And so PyMol has kind of interpolated where that loop would go, given its length. And I think it really just sort of, kind of completes a curve between that cover the space where the residues are missing and connect it to the rest of the structure where the residues are present. Let's do, back to one. Okay. One thing you can also do is, well, let's display the sequence. So, we don't have a view of the sequence down here. If I go down to the lower right and I hit the S button, now I can see the sequence up here. And in gray are the amino acids that are missing from the coordinates. And so they would be where these dashed regions are. And you can take the slider here, click on it, and move with another finger, and you get through both chains. So we've got chain A and chain B. And then at the end are the ligands. So this is that ligand in the active site. If I can click on the sequence, I can click on these amino acid sequences, and they get highlighted with these little squares. If I click on the black background again, then the selection's not actually cleared, but at least they won't be highlighted on the screen. If I go to the right-hand side now, so I clicked on, I'm going to click on I'm going to click on the sequence. And if I keep clicking on different amino acids, they actually get added. Or I can click, hold it down, and swipe with my second finger, and I get more amino acids highlighted. Whenever you've got something selected, this select object appears in the PyMol window, in the PyMol object list. And now these show, hide, label, and color and action buttons will only work on that section. So I could highlight that region and I could color it magenta. If I click on the black background again, then these are hidden, but they're still in the selection. If I took the selection on and off, it would be either highlighted or not highlighted. But if I click on the black background and then I click 4T3, now it basically cleared the selection and started over. And so the new selection is just this 4T3 molecule, this inhibitor in the active site of this kinase. And typically for a ligand, I would want to show that as sticks. And so now you can see those as sticks. And I would also want to color them. We can color it orange. Another way to color a ligand and also protein side chains is color by element. And then just pick the top thing in the element. And so now I can zoom in. And now these sticks are colored by the element type. So whatever color this object was, I made it orange, those are carbons. Nitrogens are blue. If you think there's a lot of nitrogen in the atmosphere, 78% nitrogen, and the sky is blue. That's one way to remember it. If there was an oxygen atom in here, it would be red. I'm going to click on a couple I'm going to click on the backbone here and show those sticks and then color those by element. And now I see red atoms. I see this tyrosine hydroxyl is in red. And if you think, blood is red and has oxygen, carries oxygen, it's red. Sulfur is yellow. Phosphorus, I'm just gonna forget what, I think it's sort of an orange color. And ions will be different colors. So now, let's see. The other menu here is the A menu, and there are a bunch of things that we can do with that. So I already showed you just orient. I can assign secondary structure. PyMol has its own way of assigning secondary structures. It's different than the way the PDB does it. Sometimes I actually prefer PyMol's. It just seems a little bit cleaner, but it doesn't really matter. Also under the action menu is called preset. And these are basically different ways of looking at the structure, different representations of it. So one of them is called pretty. And pretty colors it blue to red, actually from the beginning of one sequence all the way through the end of the other one, both chains. I don't like that, so I typically go back to color and by chain and chain bows so that I get both chains colored separately from blue to red. There are other things in here. So you can find polar contacts either within a selection or within the whole object. And that will draw little lines between where the atoms are that are where there might be hydrogen bonds. I don't typically like the way PyMol does that, so I'm not even going to show it to you. One useful thing here is generate and vacuum electrostatics and then protein contact potential. And I'll show you what happens. There we go. And this is an electrostatic surface of the object. I applied this to 4ZY4, so it's the whole object. And it's a vacuum potential, so it's not the most accurate electrostatic surface, but it does give you a pretty good idea. And basically where the surface is colored red, that's electronegative, usually because of negative side chains or a lot of carbonyl oxygens. And blue is positive, is a positively charged side chains like arginine and lysine, and the negative ones are aspartic acid and glutamic acid. And the white regions are generally hydrophobic or not polar. And so you can see this protein has a very dark red region there, and it's a homodimer, so here's the one on the other chain. if you did this for a DNA binding protein, for instance, you would see a blue region where the DNA binds because DNA is negatively charged. And you can click those on and off. That's what these buttons are. This charge will actually hide it. And then you have to go back to 4ZY4 to bring it back. And we can hide that little window. So I'm going to pause for a little bit, and you can try that electrostatics and some of the things underneath the A menu. Okay. As I said, you can select by swiping along the sequence. The other way you can select is actually to type commands. Pretty much anything you can do in PyMol, you can also do by typing specific commands. And so I can select SELE chain A. And it's highlighted chain A of 4ZY4. It's also highlighted chain A of 2Q0N, but 2Q0N is hidden. And now that's the selection, and I could, for instance, color that magenta. And so now chain A is colored magenta. I can select chain A and resi, which means residue numbers, 250-350. That would be the sort of N-terminal, most of the N-terminal domain. And if I click the select button, then I can see what it is I've been highlighting. And I can color that part yellow. And so if you know the residue numbers that you're interested in, because you're looking at the Unipro page or you just know some residue, you can select them by specifically on that command line. You can select by residue type, SELE res N, arginine plus lysine. So if I want to do add selections or combine them, it's one thing plus another thing, arg plus lysine. And if I do that and click on select, I see where all the arginines are and the lysines. And they're also highlighted up here in the sequence as soon as you do that selection. You have to have this select button on. So for instance, I could show those in sticks and color them blue. And so now I see where all the arginines are. And so that's a way to focus, be able to look at specific residue types. You can also select het. And het means heteroatoms, which is a term the PDB uses for ligands, anything that's not protein. I think, and not the nucleic acids, DNA or RNA. And so that's select het and turn it on. And I could show those as spheres. Here's the ligand in here. The single atoms are water molecules that the authors have modeled because they can see it and see them in the electron density. You can see just an oxygen atom. Typically, I'm not interested in those, although in an active site involved in catalysis, you may want to see them. You can hide those. If you go to the H menu, you can hide all of the waters, and then those will disappear. I'm going to go hide all of that and show it in cartoons. Back to that. And I'm going to color it back to chainbows. One thing I want to show you how to do is how to align two structures. And so remember 2Q0N is hiding over here someplace. It's a structure of the same protein of PAC1 kinase. And so what I want to do is align it to 4ZY4. There's two ways to do that. One is in the A menu for 2Q0N, there is align. And then if I move my mouse over, it says to molecule, and it basically will select another object, another protein. and I can mouse over and I've got 4ZY4, and then I hit the button. And you can see what it did. It moved 2Q0N onto 4ZY4. I'm going to hide all those spheres. And it does a sequence-based alignment first. It basically compares the sequence of the two proteins and then aligns it according to that, moves, tries to minimize the RMSD given that alignment. If there are parts of the structure that are variant after it does that, it will then remove those atoms and repeat the alignment. And you can see that up here. It's initially doing 286 atoms, and then it throws out some, and by the time you get done, you've only got 214 atoms. And so there are loops in this structure that are different between one of the proteins, one copy of the protein, and another copy of the protein. And so it's trying to align the core. It's trying to align the part that is kind of the same in the two structures. You'll also notice that in the sequence, I don't know if we'll see it. Occasionally there are regions that are, it's a little bit redundant. Now these regions in gray mean something different. They actually mean parts of the structure that aren't aligned. And so it's, in fact, if I take this, now you see there's an align object on the right-hand side. It changes the view of the sequence. So in the regular view, the gray means disordered residue. But in this alignment view, these regions are different in structure between the two and are therefore not aligned. And it will put in gaps so that you can see the alignment. I don't know that this, yeah, I'm not really seeing any in these two. But it'll align the residues that are the same in the two sequences. And then if it needs to insert a gap, it will insert gaps in one of the sequences. And you'll see that in this alignment view. The other way to do it is to align by doing a selection, comma, and then another selection. So I could say align 2Q0N, comma, 4ZY4 and ZY4 and chain B. So I'm going to select the other chain. It basically does them in the sequence order. When I did it the first time, it had two chains to pick from. It aligned it to the first chain, which was chain A. So this is align 2Q0N, comma, 4ZY4 and chain B. And now when I hit return, it's going to align it to the other protein. And it reports the RMSD up here, 2.0 angstroms. And in fact, if we do back to A, it's 0.934 angstroms. It's a lot closer to the A structure than the B. Remember, this is an asymmetric dimer. One of these has a different loop conformation. The activation loop is different. And so that's chain B, actually. Okay. I'm sort of following along the outline that's on that HTML page. And the file menu, so there's a whole bunch of menus, just like any other program. One of them is save session. And I can save this with any file name I want, but I'm going to pick 4zy4.pse. PSE stands for PyMol session file. And it will save that file. And you can see it over here on my desktop. And if I closed PyMol and then I double clicked this, PyMol would open it and it would start exactly how this looks right here. So in fact, I can do that. I can quit PyMol and double click on this, and it will open again exactly where I left off. And you might want to do that multiple times if you've got different views that you want to save. And then if I just hit save, save session, it will overwrite that if you want to do that. The other thing I want to show you how to do is to make an image. In the newer versions of PyMol, that is this draw button up here, where it says draw ray. If I hit that, it tells me how many pixels by how many pixels. If I wanted it much higher resolution, I could put a bigger number here. I could put 3,000. And it will scale the other one too, because it knows how the dimensions of this window that I currently have it in. Oops. And I can either draw fast or slow. The slow isn't that slow, so it doesn't matter unless you have a lot of structures on here. So I'm going to hit this. I'm going to hit the transparent background off, because otherwise it will give you a transparent background, which is kind of annoying if I wanted to have the background color I have on here, which is black. Or I can use white. So I'm going to hit that off and then hit the ray button. And it took a few seconds, and now it's kind of showing you what the image is going to look like. And then I just click Save Image to File and give that 4zy4.png, it's PNG format, and hit Save. If I don't click anything, I can also export the image again, and there are different formats. I don't know why, you probably wouldn't want to use them, but you could do it again, give it a different name if you want from there. As soon as I click the screen again, now the image goes away. Actually, if I click this thing again, the same image, you could save it again if you wanted to, but if you wanted to save a different image and save it to a different name, remake the image because you've moved things around, then you have to hit this back button and then do this again. Okay, I'm going to pause and let you try those things. I'm also going to pause the recording because I need to go get my charger, and I'll be back in a moment. So pause recording. Okay, and we're back. So a few more things that I want to show you in PyMol. One thing that's a command that is very useful is called split chains, split_chains for zy4. And what this is going to do is it's going to create two objects. One of them is chain A and one of them is chain B. And so for zy4 has been turned off, and now I have, these are separate objects and I can turn them on and off as I wish. One reason you might want to do that is now I can align chain B to chain A because these are separate objects and they'll move separately. So I can align for zy4_b, comma, for zy4_a. And this is going to align B onto A if I give the command that way. And now we can see where the difference is between the two structures. You can see that this loop is quite different. This is the substrate, that's B, should be B, that is sitting in the active site of the other monomer. I'm going to recenter. There's one other align command, which is called CE align. It actually behaves the other way. It's if I do for zy4a, comma, for zy4b, it's going to move the second thing onto the first thing. It does a structural alignment. If two proteins are really different in sequence, PyMol might have a really hard time aligning them. But the CE align knows what helices and sheets are and will try to align it based on those. And so the alignment should be slightly different. And yeah, it's slightly different RMSD. And so for different purposes, you might want to use one or the other. I use align just by habit, and it's quick, and CE align when for some reason it doesn't give me a good view of that. Let's just look at A here. One thing I want to show you is let's click on the ligand, which I then need to show in sticks somewhere. And I can zoom in on it. I'm going to color it white, and I'm going to color it by element. So it's got a sulfur atom there. I think. Yep. One thing that's really useful is, so I'm going to select that again, is under the A menu, there's the word modify. And so the A, and this only appears for the A menu of selections of the SALE thing listed in the select object in the list. And it says I'm going to modify this selection. And I'm going to say around residues within 5 angstroms. So I click those 3 things, and then I'm going to click on that. And now I see a whole lot of things that are highlighted. So the selection is basically changed. It's been replaced with, it's not the ligand anymore, it's all of the residues that have at least 1 atom that is within 5 angstroms of 1 atom of this ligand. And so I can show those in sticks. And now I get an idea of all of the things that are contacting it. I haven't shown you the label menu. That's the L here. So for this selection of list of residues, under L, I can label them residues. The default is to do the 3-letter code, but that actually takes up more space. And if it's crowded in the more recent versions of PyMol, you can do the 1-letter code, which is nice. So get I276 and Y346. And you can clear those. You can label different things. I mean, typically we want to label the residues, but you could label all the atom names. This didn't work. But for instance, if I click on the ligand, I could label the atom names of all of the atoms in the ligand. And so you can play with that. I realize you can label B fact. PyMol has all sorts of things in it that I've never used. But for instance, you can label the B factor. Yeah, you can actually just put long numbers on each one. I'm not sure why you'd want to do that, but it's there. So every once in a while I discover something, or my students, be working with one of my students, and she'll do something fancy on there. Like, how did you do that? Where did that come from? Another thing that is really useful is, so there are on the wizard up here, there are different wizard items. One of them is measurement. So if I click on measurement, I can now measure distances, or if I click that, I can measure angles, dihedrals, and other things, but typically we want to measure distances. And you see up on the upper left, it says, please click on the first atom. It's very polite, PyMol. And so we're going to click on, what's a good atom to click on? Let's zoom in on our ligand and our residues around our ligand. I'm going to color all of the atoms in the entire structure by element, so that these sticks for the protein are now colored red for oxygen, blue for nitrogen. And I'm going to look for a hydrogen bond. And partly because I've been doing this a long time, I can spot pretty quickly what to me looks like a hydrogen bond. Actually, I can see a couple of them. So the distance in a hydrogen bond between a nitrogen and an oxygen, or between 2 oxygens, has to be about 3 angstroms, which is 2 carbon-carbon bonds. And so a carbon-carbon bond is sort of that distance. So I'm looking at this is 1.2 angstroms. I need a distance between here and here of 3. And I bet that's going to be about 3 angstroms. There's also, in the right orientation, this NH is kind of pointed at this carbonyl, not quite from 120 degrees, the way I said yesterday, but close enough. And so it says, please click on the first atom. So I'm just going to click on the carbonyl. Now it says, please click on the second atom, because it needs 2 atoms to measure a distance. And I'm going to click on that. And it's actually a bit short. It's a 2.6 angstrom distance between those. But that's, you know, a decent hydrogen bond, actually a very good hydrogen bond. This one is also probably there to there, 2.7. Any others? Let's see any others. This one isn't so good. Like it, well, actually, no. Yeah, this one probably isn't. Well, the distance is okay, but the geometry is not very good. It's not in this NH. Well, that might be an NH2, so that might be actually okay, because the NH2 could point down that way. So that's how you measure distances. Another useful, and for each of those you get an object, and you can turn them on and off like that. And color them. I don't know if you could change the font. You can do that under the settings menu up here, or the display menu. You can delete the last object, so I can delete the last one, so now I've only got 2 of them left. I can delete them all, and when I'm done, I can click done, so that the clicking doesn't now no longer represents measuring distances. The other really useful menu here is called mutagenesis, and we're going to select a protein. And now I get a list of commands over here, and let's, the first thing I can do is I can select, it says no mutation is the default. I'm gonna select to something big, like a phenylalanine. Now, if I click on any residue, it's gonna convert that to a phenylalanine and show me the coordinates. So I'm gonna click on this threonine here. And it has built the phenylalanine side chain and put it in white. The threonine is still there. You notice that it says backbone-dependent rotamers. If I click on that, I have a choice of backbone-independent rotamers or backbone-dependent rotamers. So PyMol uses my backbone-dependent rotamer library that I showed you yesterday for the dihedral angles for all of the different rotamers for a particular residue type, and for the probability, the frequency of that rotamer. And you can see right here that it says the mutation 1.2%, 4 out of 4. And down here it says the states 4 of 4. And there are these arrow keys. So if I click the left arrow, I now see confirmation 3, and then confirmation 2, and then confirmation 1. And confirmation 1 is the most frequent one in that rotamer library for this phi psi of this residue and this residue type, which is phenylalanine. And it's 77% of the time a phenylalanine would have this confirmation. But this particular phenylalanine is buried and it's got a lot of other stuff around it. And so PyMol will show you what it's bumping into. That's what these little red disks are. That if I put it in the first confirmation, the most common one, there's a lot of red here. It is bumping into other atoms and it would not like to sit there. So PyMol kind of automatically checked all of the rotamers and picked the first one, the most probable one, that didn't have too many clashes. And so it defaulted to number 4. That's number 2, which has a lot of clashes. Number 3 also has a number of clashes. And number 4 has just a few, a little bit too close to that methionine. And so, this will help you to, make mutations inside of a protein if you're interested in a particular variant. And if I hit the apply button, it's going to actually delete the threonine and keep only the phenylalanine. So if I hit apply, the threonine now disappears. And obviously you can clear it before you apply it, and you would then can move on to another residue, and then we can hit done. Okay. I think that's about it that I need to show you in PyMol. So I'm going to pause for a little bit, and I want you to pick an amino acid, try out the measurements. Click on a couple different atoms that are represented in sticks and just see what the measurement is. You can look for hydrogen bonds, where you should see a blue atom within kind of about 3 angstroms of a red atom with the right geometry. So I'll pause for a little bit and and then try the mutagenesis menu and pick on some residue type and turn it into some other residue type. And you can flick through the rotamers, the backbone-dependent rotamers. So I lied, there were a couple more things that I wanted to show you in PyMol. First, that there's a setting menu up here where you can labels, you can change the font, cartoon, you can choose different forms of the cartoon, surface, volume. There's all sorts of ways of changing what you see on the screen. I'm not going to go through those. You can play with those. The display menu also changes what is displayed. So I can select the background and change it to white, which is typically what I use actually for papers. Smooth line. This is just a bunch of different ways of controlling it. The settings menu also has an edit all. And so you get a list of all of the different settings. So for instance, if I look for sticks, I can set the stick radius somewhere here. So right now the stick radius is 0.25. Sometimes that's a little thick and it gets too crowded. And so I could change it to 0.15. And now they're a bit narrower and it's a little bit less cluttered. The other thing was, oh, I know. When the PDB makes a biological assembly of multiple copies of the asymmetric unit, so the asymmetric unit is bigger, in the PDB file, it puts them in what are called states or models. In the old PDB format, it said something like model 1 then would have the list of coordinates for chain A, then N model, and then model 2 and the list of coordinates for chain A again, because let's say you're making several copies of chain A, and then N model. In MMSIF, I think it's on the end of the line somewhere, it'll have the model number. And that can be a pain when you're kind of, if it's all in one object. So let me show you what happens if we do that. I'm going to fetch a structure that I know is a monomer in the asymmetric unit, but is not a monomer in the biological assembly. And I'm sort of, I think it's actually the one that we looked at yesterday. HNR, comma, type equals PDB1. Now, if we look at this, there's only, maybe there's 2 proteins in there, let's see. There's 2 copies of the protein, but this structure is actually an octamer. But you notice here it says 1 out of 4. And down here it says state 1 out of 4. I can now click through the other states. And those 4 together are going to make up this octameric assembly. But that's kind of a pain. So there's 2 ways to look at the whole thing. And you want to be on the lookout for this 1 out of 4 thing. One of them is under movie, where I can say show all states. And now I can see every single chain, and I can see 8 different chains. What's actually better, and now it says star out of 4, meaning all out of 4, is the command split states, 5HNR. And now look what happens. I'm going to turn the sequences off down here. Now I've got 4 objects that are 001, 2, 3, and 4, and I can turn them on and off separately. I have to turn off all 5 H&R. Then I can turn on the first one, the second one, the third one, or the fourth one. And now these commands, I can make each one a different color to see how this structure is made up of all of its asymmetric units, each of which is a dimer. And so now you see the 4 dimers, Homo dimers, that make up this octamer. And so that can be very useful. This also happens with NMR structures. In an NMR structure, authors often solve it 20 times to basically give you 20 different models that are consistent with the experimental constraints, those distance constraints. And those come in these states, the different states. So the example I have in the web page is 6QFP. And actually you can see I've got show all states is on. If I go to show all states off, I'm only going to see one of them. And I can click through them I have to show, turn them off again. You can also hit this button, which will actually, it's the first arrow, first right arrow key. It will cycle through all 10 of those and just repeatedly. You can kind of get an idea of a little bit of the dynamics of the protein. And you can also split states on that. And now you could look at each one independently if you wanted to, and color them independently. Okay, now that's all I want to show you on PyMol. So I'm going to pause for a little bit, and you can play with those 5HNR and 6QFP and show all states, and come back when you're ready. Okay, so if you can navigate back to the HTML page, give me a second to do that. We've covered the stuff at the top, PDB web pages, the biological assemblies. I've listed a bunch of the commands that I showed you. Actually, I didn't mention this one, phi-sci. You can type phi-sci in the command window and then a selection like 1ABC and Resi 10-20. And it will give you the phi-sci values. The dihedral angles will appear in the window. Just notice that I didn't mention that. Now what I'm going to do is take you to Uniprot and show you what one of those pages looks like to get protein sequences. And then we're going to do, there's a few websites I want to show you that would do calculations on a sequence that will give you some structure information on that sequence. So the first thing it says is let's go to Uniprot. So Uniprot, I think, is the best source for protein sequence information in terms of annotations and the format. There's also NCBI, but I find Uniprots are generally better. Uniprot is divided into reviewed protein sequences and unreviewed. And you can see the numbers over here. The SwissProt is the old name for this one, a predecessor of Uniprot. And it's 560,000 sequences. These are reviewed by humans who add information about the function. They check all of the experimental information and the annotations and go and get ones that they need. Whereas 178 million sequences are just done automatically from genome sequencing. So generally the SwissProt sequences are accurate. They're not missing exons. They're correctly annotated. The Tremble sequences, the translated EMBL, translated from DNA of all of the EMBL sequences, DNA sequences, just like GenBank, are not that well annotated. Sometimes, you'll see a sequence and it's obviously missing an exon compared to, from one organism, let's say mouse, and then you look at the human protein and it's got an exon that the mouse one doesn't have. It's often because the mouse is usually well annotated, but let's say panda. The panda sequence might be missing an exon because the annotation is done automatically, computationally, and those things are not always precise. There are ways to get the full-length sequence if you look at the genome, but we're not going to go into that. In Uniprot, I'm going to just type P53 and just do a search on that, so you can look for any protein name or any phrase. And you get a list of P53 as well as other things that bind P53, anywhere where P53 was in the annotations. There are two different ways that Uniprot gives an accession code to a sequence. One of them is the number P or O or Q followed by 5 digits, which is not terribly helpful, but is precise. The other way is the old SwissProt way, which is the gene name underscore the species, where the species is always written in 5 letters. So there are kind of funny abbreviations. CanLF is dog, is Canis lupus familiaris. I don't know why they didn't just use the word dog, but they use pig, rat, mouse. So sometimes English words rather than the Latin species names, but sometimes abbreviations for the Latin species name. Like this is beluga whale is D-E-L-L-E because it's Delphinopterus lucus. I use these when referring to them and looking for them because they're easy to remember. But we're just going to go to the human entry, just hit the And we're going to look a little bit through the UniProt page to see what kind of annotations are available. And on the homework, I may ask you to go through a couple of these pages and get some experience looking through these. Over here, there's different menus where you can go to different sections of the annotation, but we'll just kind of scroll down. So you get some information about the function. I mean, at the top, you get the protein name, the gene, if it has an official gene name, and the species. And this one is reviewed, so it's going to be one of the better entries. So this is text. It's got links to publications. Then you get information about different kinds of sites. If it's an enzyme or a binding protein, there are zinc ions that are bound to P53, and here's the amino acid residues where they bind. You get different regions, like transmembrane or different domains, kinase domain. This is the DNA binding domain, is 102 to 292 of this sequence. And in fact, if I click on that, I can run blast of just that region against all of UniProt, or against, I wouldn't want to do just viruses, but against rodents. Against any particular set of sequences, I can run blast, and it will compare the sequence and find similar sequences in whatever I pick for this. I'm not going to show that. But if I scroll down a little bit, that region that I clicked on is now highlighted in yellow. So I can see in the sequence where the DNA binding region is. So I'm going to go back. Lots of things about molecular function, what the source of that annotation is, from biological processes that it's involved in, which is P53 is basically involved in almost everything. One I wanted to go to the names. So there's a names and taxonomy region, which gives kind of the official name, and then the gene name, the official recognized symbol for the gene, as well as taxonomy. Let's see, where are they? Okay. Keep scrolling down. Now we get subcellular localization, different sources for that information, publications, pathology and biotech. I've never seen it say biotech before. But this is paragraphs, descriptions of how it's involved in different diseases. Just below that are the variants. So these are natural amino acid variants that have either they're inherited or they're a somatic mutation. And so this first one, for instance, is at residue 82. There's a proline to leucine mutation in Li-Fraumeni syndrome. It's a germline mutation, and in some sporadic cancers, and then also a somatic mutation. So apparently it occurs both ways. So it'll tell you which one of those it is. If I click the residue number, it's going to show me where it is in that sequence. Here's the graphical view from the 1 to n sequence, and this is residue 82, which would be right there. And generally, there will be links to DBS&P, which has information about variants, ClinVar, which has information about disease-associated variants. There are a lot of mutations in P53 associated with cancer. So this is this gene. This is a big section. Then there's another section called mutagenesis, and these are experiments that have been performed on human P53. These may or may not correspond to natural variants that were in the previous table, but they might tell you about what happens for an active site residue. And so some of these are natural, and some of them are maybe artificial. Click down. There's a section on modified residues. So P53 is modified a lot by phosphorylation, acetylation. What else is there? Methylation. And so that again gives you the residue position, what the change is, if we know what the enzyme is that does it, then those are listed, publications, references. And so you can figure out, if you're looking at your protein, where is there a phosphorylation site that might change its activity, looking at the structure. there's a set on it, there's a region on interactions. Some of this is, well, there's a section where it's in text, just says it interacts with this or that. I wish they had links to these genes in Uniprot, and I don't know why they don't, but these should be the official gene names. So they should be easy to find. But down here, there are different databases that have binary interactions, interaction of P53 with something else. And so there's a big long list, and here are the uniprots. But in fact, it doesn't give you the protein names in this table, and that's just dumb. Sometimes I wonder whether developers actually look at their own pages. If they use them enough, they would realize that this is inconvenient. But that's what happens. Then there are some protein-to-protein interaction databases, so you can click on these. The BioGrid says that it interacts with 1,163 different proteins, but IntAct says 484. Each of these get them from different sources. Some of them are really well validated, where it requires several different sources, different kinds of experiments to determine whether there's an interaction or not. Some of them are high throughput and may have a lot of false positives. Like I bet the BioGrid has false positives, and maybe the IntAct has fewer false positives. Finally, there is a structure section, which lists all of the PDB entries where there's human P53. And it tells you what sequence region is involved in that was expressed. And you can hit the RCSB page or the PDBE page. I guess you can click and you get different, so I'm going to see the DNA binding domain. It will give you a representation, so you can click around on it. So you can quickly figure out whether there's a structure. Here's a section on regions. There were some up at the top, but this is where the different interaction regions and different domains, like kinase domain, something like that. And so it interacts with DNA, and it says 273 to 280 are responsible for that binding. It's actually probably more than that, but that may be helpful to find out. And I think that's it. Oh, then there's a section on sequences that has different splice forms. So a lot of human genes, there's one version that is maybe made most of the time in most cells, but then there are other versions that can be different. And so this is isoform, and Uniprot has picked out one that it likes, that it thinks is kind of the canonical version. Maybe the most commonly expressed, the most commonly studied. I don't really know what their criteria are. But here's isoform 2. I can get the sequence in FASTA format. So FASTA format looks like this. This is just a format for sequences. It's a greater than sign by then some kind of accession code, and then some description of it, and then the sequence, and that's it. And so you'll hear, we need the FASTA format, in fact, to put this sequence into some of the other websites that we're going to look at. And it tells you what the difference is between the canonical sequence, number 1, and this isoform 2. I think these are natural variants. So I already looked at the natural variants, but anyway, there's the variant table. Really a lot of them. One thing that is useful, I'm just going to search for the word PFAM, which is going to tell you where the protein families are in the protein domains are in this protein. So P53 that's the number for the PFAM, and there's one copy of that in this sequence. The transactivation domain, there's one copy of that. The tetramerization domain, there's one copy of that. The TAD2 is a second transactivation domain. I can click on view protein in PFAM. We're going to go to the PFAM website in a bit to put the sequence in, but and it will give this kind of bar chart of where the different domains are, and a table that says, oh, the P53 DNA binding domain here is 99 to 289, and it's sitting there. I'm going to go back to PFAM a little bit later. But this page is really long, so sometimes you have to kind of just search for a word like pfam or pdb to get the structures. If we go back to the top, actually, I'm going to pause for a second. And you can scroll through this page and look for I know I just did that really quickly. Or look up some other protein if you want, and get a look at what a UniProt page looks like. Okay. Up at the top of a UniProt page, there is the word format. And if I hit format and I say, first of all, I get a text file, which has these little 2-letter codes and then annotations that follow them. I don't use that very often, but you might want that. But what I usually click this for is to get the canonical FASTA formatted sequence. So there's P53 in FASTA format. So I'm just going to select all of that and copy it, because I'm going to go to a couple of other web pages and put this sequence, paste this sequence in, and see what information I get back. So if we go back to the HTML page, it says go to the IUPRED website, and there's a link. And a lot of, so I've mentioned in crystal structures where there's like a disordered loop, but a lot of proteins have very long regions of disorder that can be 100, 200, hundreds of amino acids long. So there can be a folded domain and then hundreds of residues of disorder and then another folded domain. And the functions of those are not always well known, but in many cases, they bind other proteins. Maybe they get phosphorylated, and then some phosphosite binding protein binds to that, and now it's in proximity to the folded domain, and maybe it does something, the two proteins do something together. It may be in there to hold the two domains apart, or to bring them together under certain conditions, like phosphorylation. They have a variety of different functions. They're disordered because they have an amino acid composition that is not consistent with folding up the protein into a 3-dimensional structure. And they've evolved to do that. They may not have enough hydrophobic residues. They usually have too many charges, too many prolines, too many glycines. There are different flavors of it that will have different properties. If you've got lots and lots of charges of one type, it will tend to extend, the disordered regions will be more extended. If you've got some hydrophobic residues, and if you have positive, negative in the charges, it might condense and actually be a fairly condensed disordered region. Still not folded, but less expanded, sort of smaller radius. So IUPRED is one of many servers that will predict where the disordered residues are based on the amino acid composition. And it's very easy to use. I just go to the window that says paste the amino acid sequence and just paste the sequence in FASTA format. And then the thing that what I usually use it for is to look for the long disorder. In other words, these big regions that are at least 30 residues, if not much longer, as opposed to short disorder, which it will look for, which is those loops in folded domains that may be just a bit too flexible to see. And then there's also an option for looking at the structured domains, which is kind of the opposite. Instead of looking for the disorder, it's looking for where the structures are. And you have to pick one of those. We're going to pick the long disorder, and I'm just going to submit. I'm going to click just, well, never mind. I'm going to hit the submit button. It's really quick. And it gives me this image of the disorder. And the disorder is this IUPRED2. It's the red line. And if it's above the line, it's predicted to be disordered. If it's below the line, it's predicted to be ordered. And in fact, you see that in the first 100 residues, most of it is predicted to be disordered. It dips a little bit below here. That doesn't necessarily mean anything. But some of the running average through this region would be well over 0.5 predicted to be disordered. Then there's a big long ordered region. In fact, it tells you that the PFAM domain is P53. So that's the folded DNA binding domain. in the middle. And then there's some disorder again. Then there's a bit of order. That is the P53 tetramer domain, which is just a single helix. It's kind of short, but IUPRED does pick up that that region should be ordered. And then it's back to disordered again. This anchor 2 thing, I don't use very often, but it's trying to tell you where in disordered regions where there are likely to be motifs or regions that other proteins might bind to. And typically it happens when this goes below the line a little bit or gets close to it. Those might be regions that bind, because of the amino acid composition, might bind an SH2 domain that, you know, binds to particularly like a phosphorylated amino acid, phosphorylated tyrosine. So I don't use it all that much. And so, you know, I might do this prediction without that blue line, because then you have to, if you're writing a paper, then you have to explain what the blue line is, and you might not be interested. Oh, it turns out you can zoom in on this too. I'm just doing 2 fingers on my trackpad and it's zooming in. So any protein that I ever start to work on, like a colleague comes to me and says, I'm working on this gene, this protein, I need to know about its structure, function. Almost the first thing I do is this disorder prediction, because I need to know where the folded domains are and where the disordered regions, because I need to think about them differently. The structured domains, I can look for structures that are homologous or identical of that protein in the PDB. The disordered ones, I might look for motifs, conserved segments that might bind other proteins, things like that. The next thing I want to show you is it says get this sequence of human MRP7. So that's the link will take you to this. MRP7 is a membrane protein. And so if you want to do a membrane protein prediction, I can close that. You can go to this TopCon's website and just paste in the sequence. And what this is going to tell us is where are there membrane spanning domains. And just hit the submit button. This will take a few maybe a little bit longer to run, so I'm going to keep talking here. And it's going to, it actually runs a bunch of different predictors of where the transmembrane regions are. This is a multi-helix, multi-transmembrane, multi-pass transmembrane protein. Nope, still working. And it's going to show us where each of those each of those transmembrane regions are, and give us kind of a consensus prediction overall. Still working. I'm just going to pause for a second, and when it is done, I will come back. Okay, the TopCons is, after a couple minutes, has not produced an answer. So I'm just going to continue, and maybe a bit later we'll see what its answer is. If we go back to that HTML page, the next thing it suggests to do is to go to the PFAM website, and we're going to put in the P53 sequence. So that is still open. I can copy that and go to the PFAM website. I have an old link. Site. Okay. And hit the search button, and I want to put in sequence. And I'm just going to paste in the sequence and hit the submit button. And it will respond with this is actually better than the link from the UniPro page, because it's the table's got more detail in it. And so here are the 4 PFAMs. Each of these PFAMs based on hidden Markov models. Hidden Markov models are statistical representations of protein sequence alignment, multiple protein sequence alignment. So what PFAM has done is it's looked at the structure and figured out, well, the P53 binding domain goes from here to here. It gets a whole lot of sequences of P53 related proteins, lines them up, and makes a model of the sequence alignment. It knows where there are conserved amino acid. In this representation, statistical representation, it knows where there are gaps, insertions in some sequences that are not present in others, or deletions. And this model you can then compare, and it's got, I don't know, 15,000 or so of these models, different protein domains. And it compares them to your input sequence and says, oh, your sequence has a domain that goes from this position to this position. So for instance, if we look at the first one is P53. That's the P53 binding domain. There's a description. It says it starts at residue 99 and ends at 289. There's a distinction between the envelope and the alignment. The alignment is where it's sure. If you click it to show the alignment, this is the alignment between the PFAM, the HMM, and the sequence. And so the HMM is represented above here. It's the most common amino acid at every position. And then if it's a match to the sequence, you will have the letters will be here. If it's just kind of similar, there'll be plus signs. But this is the alignment between, and I guess like very conserved residues. Yeah, some of them are in uppercase, some of them are lowercase. I think the more conserved they are, they're probably in uppercase. The envelope is kind of a statistical thing that it thinks it could align a little bit further. But anyway, the alignment is the one to really pay attention to. The HMM says this, the HMM length is 194. So in all of these sequences, the number of columns that have a lot of amino acids other than the gaps, that's length 194. The alignment of human P53 to this HMM went from position 2 of the HMM to position 193 of the HMM. So it basically included all of it except the first residue and the last residue. And that's typical. But if it's a really weak hit, you might, or maybe the protein is, the HMM covers, a multi-domain protein, and you only put in a sequence of one of the domains. And so you'll see that you only get a partial hit to the of the HMM to the sequence. In this column, you get the e-value, and the e-value means an e-value in BLAST, an e-value here means if I ran a sequence of this length against this database and it was some random sequence, I would expect to get this e-value number. If I get a score, this score of 205, I would expect to get 7 times 10 to the minus 61 examples of proteins that would score that high. The fact that I got one is really significant because 1 is way bigger than 7e to the minus 61, orders, that many orders of magnitude. And so you can go by the e-value. You might get weak hits, 10 to the minus 2, that you're maybe not so sure that that's a real hit. If you click on P53, it will go to the P53 family page, which will have some descriptive information about it. One of the things that it says here is clan. And if I hit the clan button, a clan in PFAM is a set of PFAM domains that are all homologous to each other. So it's a super family. And so if I go to the clan page, now you see that there are other family members, T-box, PAD-M, StatBind, I don't know what these things, runt domains, that are homologous. The structures should look somewhat alike if they're distantly related. There's other sorts of information, the species that this thing is seen in. Domain organization can be useful. It says there are 297 sequences of the following architecture, meaning there's a P53, there's the P53 tetramer, and a SAM2 domain, sterile alpha motif domain. And so these are the other kinds of domains that the P53 domain sits in sequences that have these other kinds of domains. And so you may find that helpful. You can also go to the structure list, and it will give you a list of all of the structures that have this particular PFAM. And TopCons is still not going to finish, but it must be broken. And then finally, we looked at PROTSID a little bit yesterday. The only thing I want to show you on PROTSID right now is, now that I know the PFAM, PROTSID is defined by PFAM. All of the proteins in one PFAM are listed together, and we study them, individually by each PFAM. So if I go to the PROTSID website, I can go to the Browse button and hit the letter P. I'm going to pause for a second because sometimes this gets stuck. So on that browse page, if I hit P, then I get a list of all of the PFAMs that begin with P. And I'm just going to find P53. Not that one, this one. And if I hit P53, first of all, it tells me that the clan over here, the clan ID is P53-like, but if I hit the P53 button, it's going to take a second. What it's going to show me is a list of all of the PDB entries that have P53 domain. So that includes human P63 and P73, not just P53. What's useful on here, so here's the Pdb code, is a couple of things. One is it will tell you what the architecture of the protein is. And by architecture, we mean what are the PFAM domains in order. And so some of these are P53 plus the P53 tetramerization domain, whereas most of them are just plain P53. I can actually sort by that, and we call this the chain architecture, meaning it's the list of domains in order in the chain. That's in contrast to what's called the entry architecture, which is for all of the different proteins in the entry, what is the architecture? So for instance, this structure has BRCT domains, BRCA1 C-terminal domains. It's got a chain that has 2 of those in it, and then it's got a piece of P53. And these are peptide binding domains. This piece of P53 is just a small bit of sequence, but that bit of sequence, that peptide will bind to the BRCT domains. It gives you the stoichiometry of the biological assemblies from the PDB and from PISA. And then finally, what's useful is it gives you the ligands. And so for instance, if I want to find a P53 structure that is bound to DNA, I can look in this ligand column and find, oh, these are bound to DNA, and then and these are not. And so you can sort by that. And then just look for the word DNA, and you would get the structures of P53 that are bound to DNA. And so we call this PDB fam. It's assigning PFAMs to the PDB, and it's part of ProCID. But it can be very useful if I want to look at, well, what else is P53 bound to? I can sort by the UniProt codes. And I can find, oh, P53 bound to TP53B, which is P53 binding protein. There's a structure of that. I could look for that, as well as being able to look for the PFAMs if I know what the PFAMs are, something that this interacts with. Okay. What I did is I found an old run of TopCons on MRP7, and it looks like this. So it's run these different programs to try to predict where is the disorder region. And what it says is the red line is inside the cell, the blue line is outside, and these little blocks mean transmembrane helix going from in to out in gray and from out to in white. And you see that their predictions are all pretty similar. I think it also gives you a consensus prediction. I don't see, that's not on this particular image of it, but it will give the TopCons result, which is kind of a consensus of all of these. And so this protein has 3 domains. It has a 6 here, another 6 here, and I think 1, 2, 3, 4, 5 here. Maybe, and this one might be right, might be 6 actually. So it's got, and probably kind of disordered residues in between. Okay. Finally, the last thing I want to show you is how we would model proteins. And we're going to use SwissModel to do that. There are a bunch of other ways to, online sites that will help you model proteins, but SwissModel is pretty easy to use. Good thing about SwissModel is it will try many instances to model the biological assembly. So if you've got an oligomer, it will, let's say an octamer, it will put your sequence onto all A chains and give you the octamer back, not just the monomer. That's also something that we've worked on. I'm going to do a SARS-CoV-2 protein, just because it was one that I found and might be, obviously of interest right now. If we hit that UniProt button, there is a SARS-CoV resource page. And it lists not only the proteins that SARS binds to, like ACE2 is the receptor, but also the sequences of some of these, all of the SARS proteins that are known from that genome. The one we're going to look at is ORF9b. This is may play a role in host-virus interaction. So maybe not a lot is known about it, but there is a structure of it from SARS-CoV-1 that we're going to use to make a structure prediction. So I'm just going to download the sequence in FASTA format. That's going to open it up as a text file. And I'm just going to copy the sequence. Now, if we go back to the HTML page, it says go to the Swiss model website. And then there are instructions on how to use it. If I go to the Swiss model website, it's sort of, I have an account on here, so it's got me logged in. You don't need an account, but it's helpful if you want to go back to your previous calculations. And there's a button that says start modeling, and it has something about CoV2 on there, so maybe they've already modeled some of these proteins. But I can paste the sequence in. And the first step is to search for templates. So I'm just going to hit the button that says search for templates. This will take a little while. And so what I'm going to do is I've already run this. I go back to Swiss model. And I think I have to start modeling. Yeah, it's got it's previous modeling project, so it knows that I've already run this. It's doing a search across the entire PDB. It's making a hidden Markov model, searching the PDB. Oh, actually, no, it just finished. I don't have to do that. So it found all of these templates, and it found hypothetical protein 5, which is from SARS-CoV-1. It's the same gene, the same protein. And then actually some proteins that are probably not at all related. But this template, it will tell you the sequence identity is 80%, it's pretty high. It will tell you how it's solved. It'll tell you the ligand. These are kind of quality scores. The higher the better. I think if they're well over 0.5, that's good. If they're below 0.5, these are probably all false hits. There's a link for quaternary structure that says that basically all of these structures are monomers except for this one that is a dimer. These are the biological assemblies. If I now hit build models, it's going to use this template to build the model. I could pick more than one of these. These are all from very closely related PDB entries, so I'm only going to pick one. But you could pick multiple templates, and it will actually use information from each of the templates. You might do that, for instance, if one template covered one loop and another template covered a different loop. From the alignment, this is the alignment of the one that we hit. Actually, if we go back and hit a couple of these, now you see all of the templates that I just selected and their alignment to the query sequence. And you can see that there's an insertion in the SARS-CoV-2 sequence that is not present in any of these templates. I'm not sure why this is a little bit longer here than in why these are not the same sequence. It may be that in these they're disordered. So if a residue is disordered in the template, it won't appear in this sequence. That's almost certainly what that is. I go back to templates. Now I can hit the button that says, I'm going to build a model just out of the first one and hit build models. And I don't think this takes very long. I'm going to go back to the one that I did earlier. And click on, so this is what, if I let that run, this is what would happen. Actually, no, it did run. It's that quick. So this is the one that just ran. And here's the alignment, and here's what the structure looks like. It built a monomer. And under here where it says model 01, I can download the coordinates in MMSIF format. And I'm going to choose to open that up in PyMol. And so there's the model that Swiss model built, and I can fetch the template. The template was 2CME chain B, and I can actually tell PyMol to give me 2CME chain B by just writing 2CME and the letter B. You might need to align them, but in this case I don't. They're already aligned. And you can see that here is where that insertion is. Swiss model built this longer loop when the SARS-CoV-1 protein didn't have that loop. That long of a loop is not easy to model. This is probably not right, the correct prediction. But at least it sort of tells you where it is, and you can look to see, oh, there's a big difference between CoV1 and CoV2, and that's the length of this loop, and here's where it is on the structure. I can color those, I chain those, and see where the structure is different. Tonight I will put together a homework that will have you do some things in PyMol, have you do some things from Uniprot. Maybe I put some questions in about the lecture, I'm not sure. I haven't done that in the past. And it'll be due in something like 2 weeks. We'll figure that out. So I want to thank you for sticking with me. I know this has been two-hour lectures, not 1 2-hour lecture, but when I've taught this in the past, that's the way we did it. But that was hard to fit in this semester. So effectively, I've given you what I've done in previous years, a lot more material in the lecture than I used to do. That was a biostatistics class, and this is bioinformatics, so I gave you a lot more bioinformatics, which is what I'm interested in. So I hope it was useful to you. As I said, please email me any questions. My email's the Gmail account. And I'd be happy to talk to you by video chat or phone or whatever, or just answer in an email. So please let me know if you have questions, and stay safe and stay home, and thanks for listening.